Okay, so good afternoon, everybody. Welcome to week 3 of the live interaction sessions for Introduction to Cell Biology. So, as you already know, we usually take up the previous week's assignment. Then we go through some of the questions that were there in the assignment. We go through queries that you might have regarding the same. And then we go through the topics that are for the upcoming week, which is for week 3, and end with some sample questions, which might help you in your assignments in the upcoming week. If at any point you have any queries, please do interrupt me. And if there's any noise, please, I apologize for that. The weather here is very bad, but I hope I would be audible. And please ask your queries at any point of time, interrupting me. Don't hesitate. So first, we'll go through the assignment questions that were there. So in week 3, we were asked in week 2, we were mainly dealing with topics that centered around the second part of the central dogma, which was involving topics of transcription and translation, and was ended with topics that focused on the idea of protein folding, the Ramachandran plot, and a little bit of the introduction into the possible ways in which a protein folds. So as part of this week, we'll be taking up the next topics which followed that, even though there was a little bit of overlap. We'll go through enzymes, lipids, and carbohydrates, and we'll next look into genetics, which has been explored in much greater detail in this week. So Mendelian as well as non-Mendelian genetics, and some of the basic concepts, terminologies, etc., which might be useful to somebody who is not from a genetics background or who has probably not looked into it anytime before in their academic career. So before we get into all of that, first, let us look at the assignment questions. So in the first question, you were asked that in protein translation, the first, just give me a second. The first amino acid in the polypeptide chain is always alanine, methylguanine, methionine, or glycine. So this was a pretty straightforward question. As we have discussed that AUG, which codes for methionine, is always the first amino acid that is synthesized in any polypeptide chain. It is one of the reasons the assembly of the translational complex or the translational machinery is able to occur in that way, and the correct positioning of the mRNA can occur because of the AUG codon, and therefore the first amino acid which naturally gets translated is obviously methionine. You can look up, if you're interested, you can look up at a variation of the same, which is FMET or formyl methionine, which is often seen in case of well, I'll let you Google it, but you have to look into it whether it is in prokaryotes or eukaryotes, or if there is any other bias regarding the same. So it just has a little bit of a modification, which is a formyl group that is attached to the methionine. So that was question 1, and the answer to it is obviously as indicated here. Secondly, we were asked that which of the following property is a property of introns that are present in the pre-mRNA. So if you remember, we had discussed the process of splicing, which is where introns are removed or they are spliced out, and the exons, they are ligated together to form the complete mRNA. And it is part of the post-transcriptional processing that takes place inside the cell. So by definition, what are introns? They are non-coding sequences that separate exons in pre-mRNAs, which are spliced post-transcription, which is obviously the correct answer to the question that was given here. But apart from that, let us look at the other possibilities that were suggested here. There are palindromic sequences that code for functional proteins. Absolutely not true. You will find an instance of palindromic sequences, but they have a different job. If you're interested, look up how restriction enzymes, this is a much more advanced concept, but if you're interested, read up on how restriction enzymes or restriction endonucleases, they work. There, the recognition sequences are palindromic in nature. Palindrome simply means a sequence which reads the same in either direction. So you can look it up. It's a very interesting concept, and that is explored thoroughly in molecular biology for our own needs, for our own purposes. Next, we are being asked that introns are coding sequences that code for non-functional proteins, which is absolutely not true. Introns do have other functions, but they are certainly not coding. And we can talk about if the process, if a particular process such as translation is recognizing a coding sequence, it would more often than not produce a protein which is functional. If a non-functional protein is being produced, that is harmful to the cell, and it is an energy exhaustive process for the cell as well. So it would always try to rectify any mistakes that it might have made, and would always try to make that protein functional so that its resources do not go to waste. So this would certainly not be a very feasible scenario. And whether introns are present in between exons and they aid the translation of proteins. Um, well, they are present between exons that you can consider to be true. Sorry. That you can consider to be true, but they do not have much role in translation of proteins. So that is certainly not their function, definitely not their main function. So option one is the correct answer to this. In question three, we were asked that remdesivir, an antiviral medicine that was approved for authorized use in case of emergency to treat COVID-19 in a large number of countries, which step related to viral replication in the figure below does remdesivir target? So again, this brought us back to the same familiar diagram of the central dogma that we have seen multiple times before. And here you can see that each of the different interconvertible processes have been labeled by a particular number. And among them, remdesivir particularly was interrupting the process of RNA replication, which is what we are seeing here denoted by the number 2. And this was particularly useful because of the nature of the COVID-19 virus, which was coronavirus. So it would depend if the question is altered to address a different type of virus, and the mode of action of the drug which particularly acts in that case is different. The answer would vary in each of those cases. So because this was a very common news that you could come across anywhere during the pandemic 2 years ago, 2 3 years ago, this was a rather familiar question. But if you are more interested in it, or if you are preparing for an exam which can ask you such kind of questions, you need to be a little more familiar with drugs and their antiviral drugs and their mode of action, how they potentially might work. So that was question number three. In question four, we were asked that during transcription, dash is made from dash strand of dash. So the process of transcription, like we have discussed multiple times before, if we consider them to be equivalent to a scanner, Xerox machine, etc., we used many different types of analogies. In particular, for transcription, we have to consider what is the input, what is the output, and how is the process being mediated. So transcription output is always RNA. So that would solve our first blank. The second blank is asking about what strand is addressed of which molecule. So obviously the template in that case, or the input in that case, is DNA. So blank number 3 will address DNA. And if there is any confusion regarding this strand labeling, please make sure you clear that up, because these nomenclature are completely opposite to each other. So in case of DNA, because you have two strands and transcription at a time is only taking place for one strand, therefore the way in which transcription is being carried out will actually determine how nomenclature will also be done. So you have one strand which is coding, the other strand which is non-coding, and transcription will always take place for the non-coding strand. Similarly, a different type of nomenclature exists for sense and non-sense. Switch on. Yeah. So a different type of nomenclature also exists for sense and nonsense strands. So you can look into that as well. And please make sure that whenever you are addressing questions, you are making sure that these individual nomenclatures you do not mess up. So yeah, for the correct answer for this particular question would be option one, where RNA is the output and that is being made from the nonsense strand of DNA. So the next question, I think we had taken up a similar problem in the sample questions that we looked at. So if you look at the electron microscopy image, you can see that a certain lariat-like structure, a loop-like structure is being formed during this process. And the process is obviously representative of something that is going on inside the cell. So we were asked what process is it, and among them we had replication, splicing, transcription, and protein synthesis or translation as the options. So certainly among them, the correct option is splicing. So in question 6, we were asked which of the following is not a property of the genetic code. So again, this was something we took up in a lot of detail in the last week. So among the different properties of genetic code, we saw that there is degeneracy in the genetic code, but you can see that several different amino acids can be encoded for by different codes. So you can have many different genetic codes or many different codons, triplet codons, which code for the same amino acid, which is indicative of how degenerate it is, which is why you have more number of codons than you have the number of naturally occurring amino acids. So that is 64 versus 20. So that validates the degeneracy of the genetic code, which exists uh, in nature or within the cell. And universality is also considered to be one property of the genetic code, and which is why we can say that the first option is not correct. But at the same time, we have also seen that there are always some exceptions to this process as well. And a primary example of that is that within the mitochondria, we can often see that some of the stop codons or nonsense codons, which we see for other like in case of the normal genetic code, they are coding in case of the mitochondrial amino acids. And also there are other exceptions to this. You can look up if you're interested. So universality, while is true, there are exceptions to it, but at the same time, we cannot say that each species in the living world has a different genetic code addressed to it. That is not true. So therefore, option 1, which you can see, that is not correct, and therefore would be the answer to our question here. So that's what we see here. And in question 7, we were asked that which of the following amino acids are an exception to the Ramachandran plot. So we discussed the Ramachandran plot in the last week, where where we saw how the phi and psi angles, which are the dihedral angles for proteins, they are plotted against each other from -180 to +180 to show what are the possible conformations that each amino acid can potentially take um, within a living system. And among them, we saw that while most of them have some constrained regions, we saw some exceptions in terms of glycine and proline and where they could occur. And that was simply because of their chemistry, because of the structure that they possess. Because proline has a more restricted structure, it has some definite regions in which it can occur. And because glycine is has a very short side chain, and it simply just has a hydrogen as its substituent group. Therefore, it has a lot more freedom, and therefore it can have a lot. It has more number of possible conformations in which it can occur, which is represented in the Ramachandran plot that we talked about. So that is the exception we are referring to here. In question 8, we are being asked that select the sequence generated when the given coding sequence of DNA is getting transcribed. So this can be a tricky question, which is why I said that you should have the terminologies very clear in your head about coding, non-coding, sense and nonsense. So when coding sequence is being mentioned, coding means the strand whose code is exactly generated or is exactly recapitulated in the mRNA. That is the definition of it being the coding strand. Therefore, if you are being if you're being given the sequence of the coding strand, that is directly the sequence which you'll get in the mRNA and in that exact orientation as well. So this is a very simple question to address just if you have your terminology straight. So basically the sequence which is being given to you, the coding sequence, is exactly what you will get after transcription has been carried out, which is why the first option is correct. Then in question 9, we were asked about the secondary structures of proteins, about how alpha helices and beta pleated sheets, they are formed in which structure of protein. So we saw that there is some, not exactly hierarchical, but you can see that there is a process in which protein structure is ultimately being formed. So it starts off with being a simple stretch of amino acids, which is the primary structure. Then it assumes small regions where some amount of folding or some amount of arrangement has taken place. And that is what gives rise to the secondary structures, which we studied are the alpha helices and beta sheets, which is what is the answer to the question. And beyond that, you see there are tertiary structures, where these secondary structures, they assume some specific motifs or some specific arrangements, which this will go into too much detail, but yeah, you see some motifs using some domains, and then each of these, they assemble to form the final functional form of the protein, which is often the result of assembly of many different subunits or many different domains, ultimately resulting in the tertiary structure, which an example of which was hemoglobin or myoglobin, where multiple chains they assemble together to form the final quaternary structure, which is capable of performing the function that it is destined to perform. So this is the way in which the arrangement actually occurs, and you can read up on it in more detail. There is a lot that goes into understanding of structural biology, but yeah, these were the basic ideas that were discussed, and that kind of answers your question as well, that alpha helix and beta pleated sheets are part of the secondary structure that we talked about. Yeah. And in question 10, we were asked that which of the following options correctly label the peptide date. So in question 10, we were asked that which of the following options correctly labels the peptide torsion angles that are shown in the adjoining figure. So we talked about the dihedral angles in the last class as well. So you can see that this is the CONH or the peptide bond that is being shown here. And with respect to that, we are seeing three different angles being displayed here. So one direct question that you can answer from here is that the peptide bond by itself, which we have established, is planar in nature, that has been described as the omega angle. So the third labeled angle is the omega angle. And then you look at the other two options. So there is CC alpha and there is NC alpha. So CC alpha is being represented as psi, while NC alpha would be represented as phi. And therefore, the third option given here would be the correct option for your assignment question. So that basically wraps up what was discussed in the previous class and what pertains to the syllabus from that time as well. So now I'll come to the topics that are of interest for this week, which is in week 3. So in week 3, there is, as I was saying in the beginning of the class as well, what was initially looked at, what was initially looked at was overlapping with the topics covered in the previous weeks as well, about protein structure, its folding, the protein folding problem, etc. So, excuse me. So the topic that we will be starting from today are enzymes. So again, if anybody has any doubts, please do interrupt me. Just to Okay, so we are talking about enzymes here. So what are enzymes if we talk about that? Enzymes basically are protein molecules. So we talked about the central dogma in a lot of detail, right? We talked about how they work, how each of the different steps within the central dogma, they work to ultimately produce proteins that are considered to be the functional molecules within the cell. And the reason they are considered to be the functional molecule is that apart from regulating all of the different processes that we have talked about, they are also extremely crucial for making sure that each of the different processes are synchronized with each other. So there are proteins involved. If you think about it in depth, all of the different replication machinery, the transcription or translation machinery is heavily dependent upon proteins within the cell. And each of these proteins in these processes, they are all enzymes. Talk about polymerase, talk about a ligase, talk about helicase. They are all enzymes. So what is their job? What do you understand by enzymes is the question that comes first to your mind. So enzymes, okay, here we're again getting water as the option. So I'll just explain this via speech. So enzymes basically are, like I said, they're protein molecules, and their job is to accelerate um, the speed at which a particular reaction is taking place. So there is another terminology which any of you from a little bit of a chemistry background would also be familiar with, which is that catalyst. Okay, just there's something wrong in this presentation mode. Yeah, so another terminology that we were talking about are catalysts. So catalysts, as the name it indicates, it catalyzes a particular process. So you have some initial reactants, and then you have products which are made out of those reactants, and the mediator of these processes is basically a catalyst. These catalysts, which work within a living system and which perform very difficult tasks of taking these reactants inside the cell and carrying out processes which under normal circumstances would take years to complete, and they execute that action within a matter of a few microseconds, picoseconds, etc. The time scale would vary depending upon what kind of process we are talking about. So they perform this extremely crucial function of converting these reactants or carrying out these particular set of reactions. Like, for example, if you just take the instance DNTPs, and it is able to synthesize a polynucleotide chain within an insanely short amount of time. Therefore, the job of the enzyme is basically to carry out all of these processes. It assembles the reactants very close to each other. It provides a sort of scaffold. By scaffold, I mean a sort of surface where each of these can assemble, and then it can carry out, it can provide a space where these reactions, they can take place in a very defined, very limited amount of time. And then it produces the product, which is the job of the enzyme that it is supposed to execute anyway. So this is what it does. And now enzymology is again a entirely different topic that if you are interested in, you have to get in a lot of detail. There are enzyme kinetics that are looked at, the speed at which an enzyme can perform a reaction, the ways in which an enzyme can function. So there are several different hypotheses or different ideas which have been proposed as to how an enzyme exactly does it. One of the most common ways is to go via a lock and key mechanism, where it basically allows, it locks them in a certain way, and it converts the it allows the reaction between each of these reactants to proceed. It produces the product, and then it releases it. So there are many different ways of doing it, and you can look into it if you are interested. So that is an overview of how exactly enzymes might work inside the cell. Following that, we are looking at water. Sorry, this was part of the previous week. We'll now come to lipids, which was the next topic that was taken up as part of this week. So lipids, as you can see, they basically have two different so lipids basically have two different regions. They have some water loving and some water, not hating, but afraid of water regions, so hydrophilic and hydrophobic regions, which you can see here. And this is an example of an individual lipid. It has this is the example, or rather I could say, of a phospholipid, where it has a head region which is water loving or which is hydrophilic in nature, and a tail region which is hydrophobic in nature. And you can see that within the living system, there are a huge variation. There is a huge variation in the types of lipids that exist. Like there are glycerolipids, glycerophospholipids, sphingolipids, etc. Each of them have their own function. But when we are talking in context of cell biology, they are extremely important because if you think about it, when we talk about organelles, when we talk about a cell, one of the major defining characteristics that would establish what a cell is the outer membrane, right? It is the boundary which is separating one cell from another cell or from its external environment. So that is being taken care of by the plasma membrane or by the cell membrane, which is entirely made up of lipids. So this membranous structure, as you might already be familiar with it, is said to be a phospholipid bilayer. What does that mean? It means there are two different layers, each of which are made of phospholipids, and these are surrounding the entire cytoplasm, and they are constituting what exactly a cell is. So this bilayer is made in such a way that the outer surface of each of these two layers is made up of phospholipids arranged in a manner that their head region faces outside, while the inside is made up of the phospholipid tails. So that is what you can see here as well. You can see the color is not very distinct, but what you'll be still able to see is that you can see there is a distinction between the two regions that are present. And this representative example is much easier to understand. So this is a phosphatidylcholine. Basically, these are phosphoglycerides or lipid molecules, which make up one of the lipid molecules which make up the cell membrane. So it has this hydrophobic non-polar tail, and these are made up of fatty acids, and it has a glycerol backbone to which it has these some specific substituent groups known as phosphates and cholines attached to it. And these are forming the head group that we talked about, and these are the water fearing regions or the hydrophobic regions, which are always restricted towards the inside of the bilayer. It is not exposed to either the cell environment or to the exterior. So that is what we see here. And here you can see these are representations of the lipid structure. And you can see fatty acids, glycerolipids, glycerophospholipids, sphingolipids, sterols, and prenols. So most commonly, glycerophospholipids, sphingolipids, and sterol lipids, you can see that they are some of the major contributors of the cell membrane. Among them, phospholipids are the uh, how should I say, the maximum constitution or major constitution of the membrane is actually made up of phospholipids, glycerophospholipids in particular. And these are present in some specific regions and they have some specific functions. Then there was a video on lipids, which we are not getting into. But yeah, if you want to study more about lipids, there is again, each of these topics are just introductory in nature. You can get into a lot more detail about them. But lipid biology is extensive. It's not just the outer cell membrane. There are intracellular membranes also. For example, the endoplasmic reticulum is made up of membranes. The nucleus also has a membrane which has a completely different nature because it has little pores. Because as you remember, we talked about, sorry, is unmuted. Because it's going, yeah. Because we talked about how after the process of transcription, you have to export that particular mRNA which has been synthesized from the nucleus to the cytosol. And for that to be allowed or for that to be carried out, there have to be some exit channels or some gates, you can say, within the nucleus boundaries. So those are formed by the nuclear membrane itself, and there is another complex machinery which operates there as well. So without getting into that much detail about it, what you can understand is that the lipid, even within the cell, lipids of different regions have different compositions. They have different compositions and they have different roles. And one example or you can, not an exception, but one particular instance where a lipid is present and has a different function within, not within, but rather on the outside of a cell is for neurons. So if you've studied a little bit about neuronal structure, you would know that it has a sort of covering, which is known as the myelin sheath. So a neuronal cell individually, if you represent it, it has some dendrites, it has axons, it has a cell body, and it has a long stretch in between, which is covered by a certain coating. You can think of it in that way. And that coating is made up of some certain types of lipids, which are necessary for the neuron to be in its proper health. And when these lipids are lacking, I don't know if you're familiar with it, but or if you've heard of the disease known as multiple sclerosis, which is an extremely serious disease, a neurodegenerative disease, which is caused because this myelin sheath, which is the coating of lipids around a neuron, when that doesn't function properly, because that is not, the neuron is not coated properly, it needs to misfiring of neurons. They're not able to communicate properly, and there is a complete like discoordinated or uncoordinated signaling that goes on within the brain. So neurons are not able to convey information properly or relay information properly to each other, and such a person starts behaving very abnormally. And within a very short time, a few years, when I say short time, I mean in terms of their entire lifespan. Once a person suffers from it, they die very soon. It's multiple sclerosis, or rather, most neurodegenerative disease don't really have a cure. So that is just an example that I was trying to bring to your attention how important lipids are. And apart from that, you also have the fact that a lot of people, if you're familiar with it, they suffer from fatty liver disease. That's because of a number of other reasons, but also because lipid metabolism in such individuals gets affected. So you can get into details about this as much as you want. But yes, they are another very, very important major macromolecule within the cell. Not just proteins are important, but equally important are lipids, and also the next part that we'll be coming to, which are carbohydrates. So carbohydrates, as the name indicates directly, it means hydrates of carbon. So they have carbon molecules and they have multiple hydrogen and oxygen molecules, which can be represented in terms of the number of molecules of water, which you can see here. Um, Sorry, this is a representation of the same, and you can see different projections of different types of carbohydrates that are commonly found within the cell. We have glyceraldehyde, dihydroxyacetone, sorry, fructose, ribose, etc. So as you can see, these are all images that are taken from Leninger. If I have not mentioned already, I think I have, but one of the best books you can go to for understanding the basics of biochemistry is actually Leninger. You can refer to other books as well, Voigt and Voigt, etc. These are all really wonderful books, but for basic biology and for introduction to what exactly is going on inside the cell, please go through Leninger. It will not help you just today, but in the long run, especially if you're interested in academia. So please refer to that book. It is wonderful. So here you can see these different examples of carbohydrates within the cell. You can see the representation of starch or amylose, which is a slightly more complex carbohydrate than what you see here. Again, there was a video here that was talked about, and we can see different polymers, carbohydrate polymers. When we say polymers, we mean multiple sugar moieties that are linked to each other. Like, for example, there are disaccharides, polysaccharides such as hyaluronate, chondroitin sulfate, keratin sulfate, heparin. These are all parts of the extracellular matrix. So apart from the cell is not just lying in isolation, right? There is something surrounding the cell or the cellular environment, and there are certain um, connections that are made with the cellular environment as well. These are all molecules, examples of such molecules, which are part of the environment, otherwise known as ECM or extracellular matrix. And these are some of the components of it. And the cell has to interact or has to form. Think of it as a bed. Think of it as you lying on a bed. If you are a cell and you are resting on your bed, and that is the substratum on which you are kind of dependent in a large way for your rest, for your functioning, for you to be fit to work the next day, then the ECM serves as the bed on which a cell is laid. So it is present around the cell, and it is necessary for the cell to perform its function and for it to survive the way it does. In fact, one of the major ways in which or one of the major transitions that take place when a cell is set to become cancerous is that its connections with this ECM, they are completely disrupted. And as a result of that, it is no longer lying on that bed, right? It is ready to go about and do whatever it wants to do anywhere in the house. So that is equivalent to a cancerous cell losing its initial address and it becoming free to metastasize to any part of the body and cause a lot of havoc. So this is just an extrapolation of the idea. So that is what you see here as carbohydrates, and these carbohydrates can be conjugated to proteins and lipids as well. They can be thought of as little decorations, and they are necessary particularly for bacterial cells. You will find a lot of such decorations on bacterial cell walls. So they have their individual functions. You can look it up if you're interested. Please do interrupt me if there are any doubts at any point of time. Following this, in the next lecture, we were introduced to the idea of what is genetics. So I'll come to that. Yeah. So the initial lectures, they talked about Mendelian genetics. So as you already know, Gregor Mendel was well, he wasn't really a scientist. He was more of a naturalist who was exploring, he was observing what was going on around him and was trying to make conclusions based on it. So what he had initially observed was that he was looking at pea plants and he was seeing that there is some sort of a pattern or some sort of a linkage which he is able to see in what were the parental plants and what is the next generation. Is able to link them somehow, and from there he drew some conclusions which, while they were not initially accepted, over time they were studied in a lot of depth, and it was eventually understood that yes, all those decades ago Mendel was indeed right, and what he had said initially did make sense. And then those theories got established further, and we could even observe that there was a lot of deviation from what Mendel had proposed, which is why we get into the idea of non-Mendelian genetics. We come into the idea of how complex genetics is, and it is um, not just the genetics which can influence what the next generation would look like, but in fact the genetics and the environmental factors which altogether decide what a particular individual, what traits a particular individual would have in the upcoming generations. So there is a lot that gets contributed to it, and we'll not get into that much detail, but for now what we'll look at are the ideas that were brought forth by Mendel at that point of time. So, as I said, he had carried out a lot of experiments with plant hybridization. So, hybridization, again, as was mentioned in the lecture as well, while when we talk about biological macromolecules like DNA or basically nucleic acids or even a protein nucleic acid interaction, what we mean is that they are two molecules which are binding to each other, which are interacting with each other and forming a new complex. But when we talk about plants, it basically means they're crossing over, which means they are it is equivalent to animals which are bred with each other or which are mated with each other. And the generation which is derived from them is what is representative of characters from both. So that is the example that he was also intending to study. So he did a large number, uh, unimaginably large number of crosses were carried out to understand how exactly information carried in one generation is being taken forward to the next. So it was a long and laborious process, but it gave us some of the basic laws brought forth by Mendel, some of the basic ideas, which of course then got explored further to validate them. But let us look at what exactly he studied. So, according to Mendelian genetics, what he came forth with was that he developed a set of rules based on which he concluded that this is how hereditary information is carried forward. This is what heredity means, and how is information from one generation being taken forward to the next. And his observations were not based on experiments as we might think of them now. For example, if you think about um, the semi-conservative nature of DNA replication, the way it was studied, I'm not sure if you're familiar with it, but there were a lot of very eminent scientists who carried out a set of very brilliant but very simple experiments where they kind of understood that if there are two strands and you label them differently, then in the next generation you get one retention of the parental strand and one strand with the new labeling being incorporated. So you can look up these experiments. These are some of the most landmark experiments you can go into for molecular biology, Messels and Stahl's experiment, and many more. I'll not get into detail of it. It's beyond the scope of this course. But those were very targeted experiments at trying to understand what exactly is happening with DNA inside a cell. But in case of Mendel, he simply carried out crosses between plants that had some differences in their traits, and he tried to conclude what he observed based on these experiments. Yeah. So that was the goal. So based on these simple observations, what he tried to understand was whether there was a ratio or whether there was a pattern that was being retained, and whether it was statistically significant when it is performed for a large number of plants. So this he did across many generations of the pea plants. However, on the other hand, non-Mendelian genetics, these were slight deviations from it. Mendel's rules were based on the systems that he had used, and they were not generic in nature. Some of them had to be modified, and therefore we see some deviations from it. So this you see Gregor Johann Mendel with his other monks, who were part of the same, uh, well, for lack of a better word, fraternity. But then he had uh, carried out some exceptional observations, and he is cited. He was recognized for his work and was cited much later. So the basic idea behind it is that the cellular genome, it has all of the information that is required to make a new organism. How that organism would eventually evolve to behave and to function is dependent on a number of other things. It is dependent certainly upon the environment, which is what I talked about, is the concept that has come up much later. But just the information within a cell is capable of forming a new cell, and from there an entire organism. So that entire information is being retained within the genetic material of that individual, which is that individual's genome. So that is what we talked about. And this, you can see that approximately 3 billion base pairs that are present within the human genome, they are all being recognized chromosomal, into the chromosomes, or they are being organized into the chromosomes at the time of cell division. And we know that for humans, which are, of course, diploid organisms, we see that there are 21 set of autosomes, and there are sex chromosomes apart from that. So 22 pairs of autosomes and sex chromosomes apart from that. So total 23 sets of chromosomes can be seen, and you can see that these are XX or XY, which determine the sex of the individual. And any sort of um, non-dis okay, these are two terminal, but any sort of uh, deviation from this normal chromosomal architecture can lead to a lot of diseases, like I think we discussed previously as well, such as trisomy of 21, Turner syndrome, etc. A lot of diseases occur when chromosomal arrangement or chromosomal architecture is disrupted from one generation to the next. So this is the basic idea of how chromosomes are arranged. And here, in this slide, what we understand is that these information that we talk about, that the genome contains, is stored in pairs of files. Like you could see here, each file is a double-stranded DNA molecule. So the analogy that was given here was that if you think of it as a hard drive, you will get, because humans are diploid in nature. Diploid basically means we have two copies of each information. We have one, you can think of it as backup in a way that you have one main file and you have one backup file. So there is chromosome 1 text and there is chromosome 1 prime text, which is sort of a backup of the information that was there in chromosome 1. And similarly, you see it for all of the chromosomes that are present. So these are the information that is being shown, or that is being represented with respect to the content of each chromosome. You can see that all the 22 chromosomes and the sex chromosomes are being represented along the x-axis, as you can see here, and the total number of genes. So the number of genes kind of reflects, while it is not an exact measure, but the number of genes reflect how much information is contained within these chromosomes. So you can see that chromosome 1 has a large number of genes, while the number of base pairs is not that different from that of chromosome 2. So this is why I said that the amount of information does not depend on how much or how long a chromosome is, or how many sequences you can actually, or how many base pairs you can recognize within it. So while the number of base pairs might be quite similar in a large number of chromosomes, the amount of information that can be derived from it, or the number of genes that are present or coded within that chromosome, that can very significantly vary. So you can see that there is a huge variation in the number of genes encoded, particularly if you look at chromosome 18, while it has a large number of base pairs, the number of genes in it are quite less. Versus if you look at Y chromosome, it is quite small compared to the X chromosome. So these are all very different. That brings about a different idea that males, they have one X and one Y chromosome, but females have two copies of X chromosome with so much more information. So what happens there? So there are many cellular mechanisms which come in. I think it is beyond the scope, but if you're interested, you can look up the term dosage compensation, which occurs in females. Basically, because we have two copies, females, they have two copies of the X chromosome. One of the chromosomes is basically wrapped up. It is just, think of it as wrapping it up in foil, so that whatever is inside that chromosome, whatever information is encoded inside that chromosome, does not get converted into functional proteins. So we have effectively, females have the same amount of information as males do. So these are very complicated mechanisms that occur within the cell. Just to get you a little bit introduced to the idea, I mentioned it, but certainly it is a very advanced concept that has a lot of significance. There is a reason why dosage compensation occurs. There are a lot of consequences of that process as well. But for now, this is what you can just take as a source of, just as an anecdote or as a source of information. So yeah, so we were talking about how each file is a double-stranded DNA molecule. Then each of these terms they are very important concepts as part of Mendelian genetics. So what is a gene? What is an allele? As was initially discussed, I think in the first interaction session, somebody had tried to clarify what is a chromatid versus what is a gene. So make sure your ideas about each of these terminologies is very clear. What is a gene? What is a chromosome? What is a chromatid? What is a homologous chromosome? Or what are homologous chromosomes? What is a trait? And each of these definitions, they are important because that is based on what Mendel had, based on these definitions, Mendel had kind of laid down his whole idea. So if you want to understand Mendelian genetics, you should know what he meant by these things. So please make sure you have clarity about these concepts. So however, we'll just go over the basic idea. So like you can see here, these are the individual chromatids, which are part of the same part of the same chromosome. They are attached at the centromere. And when replication occurs, they form two pairs of homologous chromosomes, and these individual are known as sister chromatids. So next we look at the different characteristics or different traits that Mendel had actually looked into. So he had looked into, so when we talk about the fact that Mendel had done a lot of extensive crosses or hybridization among pea plants, and he had studied them to see whether they were statistically significant, what would he study? So you can allow two plants to breed with each other. You can control the breeding between them. But then what will you study? You always need something visual, some visual characteristics to conclude that, okay, plant X and plant Y were bred with each other, and plant Z, say, which is a product or is the progeny of plant X and plant Y, will have these characters. So for that, you need to have certain traits, certain observable traits, which are otherwise known as phenotypic traits. What you can visually see is a phenotype. What is contained within that individual's cells, the information within that individual cells is basically the genotype. So when Mendel was studying, he tried to find the correlation between these two, the phenotype and the genotype, and he tried to draw conclusions based on that. So that is basically what we look at. So these visual characteristics or these visual traits that we talked about are, as has been shown here, he looked at the seed coat color, or he looked at the flower color, whether they were gray purple or whether they were white and white. The seed color that is within the pea plant, what is the color of the seed, whether it is yellow or green, whether it is whether it has a distinctive shape, whether it is smooth or wrinkled, the color of the pod shaped, pod shape, and the stem height, and finally the flower position. These were the initial characteristics that were looked at. And particularly what he looked at in great detail was the stem height. It was extensively studied by him, the tall, and it is also the most commonly cited example when it comes to Mendelian genetics. That is, he looked at the height of a plant, whether it was tall or short, and how that had changed over generations. So you can you can also try and look at that, or you can cite that as an example for your use as well. So here you can see the kind of representation that he showed, and the relationship between the modern terminologies that we talk about with respect to genetics, and back then in the 1800s, how he had used these terminologies and how he had labeled them. So you can see here the different characteristics and how they are represented: the seed color, coat and flower colors, the surface or the texture of the pods, the inflorescence, which is the location of the flowers, the height of the plant, and the shape of the seeds, the color of the seeds, etc. He all represented them in the in his own ways, and they are represented differently under current circumstances. So this is a way that shows how he had exactly done the hybridization process, where he used to extract the pollen from one plant, and he used to cut away the anthers of the plant itself, and then he brushed the pollen from the other plant onto the other recipient plant. So basically, the easier way to think of it is that you take two parents, you take one of them as the male, the other as the female, and you take the anthers from the male plant that you want, whose traits you want, and you brush those anthers onto the pistil, or onto the female reproductive parts of the flowers from the plant that you want to be the female parent. So this you do, and you then wrap that plant with some sort of a plastic bag or some sort of a covering, so that no external pollens can come onto it, and therefore only what you have allowed, only that cross pollination would be able to survive, and then you would be able to repeat this over multiple generations. This is just one generation where you get a mixture of genetic information. When such information is carried out, or such crossing is carried out over multiple generations, what you effectively end up getting is sort of a when it is between similar traits, what you end up getting is a pure breed. Like for example, if you take a tall plant as a male and a tall plant as a female, and you cross them, and you cross them for say 8, 9 generations, say 10 generations, you cross them. What you expect to see is that the progeny that you see as tall in the 10th generation is pure breed, and it carries only the genes which are necessary to become tall. So these are the ways in which plant hybridization is carried out. So here you can see that these are particularly important for diploid plants, which are which carry out sexual reproduction. They have a large number of seeds, and they can be self-fertilized or cross-fertilized, depending upon what is our objective. You can also carry out self-fertilization to produce similar plants. But if you want to see crossing, or if you want to understand how genetics is getting, or how traits are getting inherited across generations, then it always makes sense to carry out cross-pollination among different plants. So that is the concept that I just explained. And in this image, what you see here, let me just zoom it a little bit. Uh, Basically, what you see here is a way in which chromosomes are studied. There are many different techniques for visualizing chromosomes. You can see them just under the microscope. In fact, if you have some background with respect to biology in your, mostly in your senior secondary, usually there are some experiments that are done with onion root tip cells, which are easy to squash, and you can do karyotyping for those cells, and you can see the different stages of cell cycle and how they are arranged. You can see individual chromosomes in different formats. Sometimes they are arranged along the metaphase plate, sometimes they're separating in the anaphase, or they can be arranged in as just as a as chromatin in a non-condensed format. So all of that can be studied, but in particular with the advancements in technology that have occurred in the last few years, one of the methods for studying chromosomes or visualizing chromosomes has been FISH, FISH, which is fluorescence in situ hybridization. Um, What that basically means is that you are fluorescently labeling or tagging chromosomes inside the cell. Let's just keep it at that. So here you can see the representation of each of these chromosomes. You're using certain markers which are allowing you to specifically highlight one portion of the or one particular chromosome and one particular portion of one chromosome. So that is what you can see there as a representative example for each of these different chromosomes. And these are markers, the red and green markers that you see, they are specifically located on these chromosomes, which is why you're able to visualize them. So yeah, that was the idea that was explored by them. Now we come to the conclusions that Mendel had basically drawn. So Mendel had a couple of postulates that we talk about. So what did he say? First, he saw that unit factors, he called genes or alleles as unit factors. He saw that genetic characteristics, they are controlled by unit factors that are present in pairs. So these unit factors in current terminologies are commonly understood as alleles, which means that each individual, each diploid organism, they have an allelic pair which is represented or which is present on chromosomes, and this allelic pair is present on you can think of them as homologous chromosomes within the nucleus of an individual cell within an individual organism. So he said that these unit factors do not mix or do not contaminate with each other. Do not contaminate each other. They are located on distinct sets of chromosomes, which is correct. Basically, what he said was that genes are present in two associated copies in diploid organisms. So his idea in the current scenario basically means there are two chromosomes which are homologous. When we say homologous, okay, another question that might come up is why don't we call them identical chromosomes? Why are they not the exact same? That is because if you think about identity, it means if you are just xeroxing a piece of information, say you have a paper which has a particular paragraph, you just xerox it, not a paragraph, say a book. If you have a book and you Xerox that book, what you get as the output is the exact replica of that book. Now, say you have a book and you are somebody and a human being is writing whatever is written in that book by hand. What do you expect? You expect there to be some errors, you expect there to be some changes, which are not intentional, but it is leading to some variation from what was there in the original copy. So what you have now is similar to what was there initially, but it is not the same. That is exactly what we mean when we say homologous chromosomes, which means they are quite similar, but they are not replicas of each other. This variation or this dissimilarity, it comes because the idea of unit factors that we just talked about, in other words, they are known as alleles. Alleles are different, very slightly differing versions of the same trait. We'll come to more details about it. Say, for example, when we talk about, okay, let me just give you an example of somebody's eye color or hair color. Let's talk about hair. These are, don't use these examples because these are multiple allelic traits. They have many different alleles in many different regions which code for it. They're polygenic traits also. Don't go into details of it, just for your understanding. Say you take somebody's height as an example or somebody's hair color as an example. Say one of their parents has brown hair color, the other parent has is blonde, has golden or whitish golden hair. That their child, whoever is their child, will receive one copy of each of these. What will happen effectively is that their hair color, this child's hair color, can be determined by either of these two, by both of these two, or can be a mixture of these two in different ratios. So that is what we are trying to say, is that while both the alleles on two different chromosomes, they have received two copies from each of the two parents. Say, for example, we are talking about one particular site. Say within the chromosome, you have one particular region which is called as P14. Say, for example, then P14 on chromosome 1 and copy of chromosome 1, one is maternal, one is paternal. On each of them, P14 is encoding for their hair color. Correct? But each of these, the information within those regions is different. It is slightly different, but it is different. And therefore, what the final child's hair color would be determined by what is the information encoded in them. I hope I got the concept across, which is why we say that these two chromosomes, while they are similar, they are not exactly the same, and therefore they're called as homologous chromosomes. Now, what will be the trait is determined by a number of complex factors. We'll not go into detail about it because it depends upon Mendelian and non-Mendelian genetics. Some traits or some characters are simply decided by Mendelian genetics. Some are decided by a group of genes. Some are decided by genes as well as environmental factors. There's a lot that goes into it. So we'll come to that later. So the idea that I was trying to introduce was that unit factors are usually present in pairs. They do not contaminate each other. They are present in two copies when we talk about diploid organisms. For example, the plants, when we talk about their stem height or we talk about their height in general, they have two different variations that have been mentioned here. Capital D plants have two alleles for tallness, whereas small d plants have two alleles for dwarfism. The second concept that he brought about was the idea of dominance or recessiveness. What he said was that uh, unit factors, unlike in the case of when unit factors are not the same, that is when they are not capital D or small d, but they are a mixture, which is simply known as, in the current terminology, they are known as heterozygous conditions. Heterozygous means different conditions. They're just different. The zygosity is different. Homozygosity means they're the same. So under heterozygous conditions, he proposed that there would be one particular allele which is dominating over the other. This is according to Mendelian genetics. Keep in mind, there are, of course, exceptions to this. There is incomplete dominance, there is multiple allelism, and there are so many other variations of this. But his observations were simply based on what he saw in pea plants, right? So he said that one can be dominant, one can be recessive, and therefore, in other words, he said that when two different alleles of a gene are present, one may show its effect while the other may be masked. So the one which is showing its effect is obviously dominant. The one which is sort of getting masked is the one which is recessive, right? So, for example, the capital T small d heterozygous plants have a tall allele and a dwarf allele, but they are phenotypically tall. So despite what the genotype might be, which is that they have a mixture of both alleles, what ultimately you are able to see is that the plant is tall. You have to figure out whether it is a capital D homozygous condition or a capital D small d heterozygous condition. Then comes the idea of segregation. So according to Mendel, and keep in mind, this is according to Mendel, he said that the formation of gametes, which is the process which takes place when an individual within an individual by the process of meiosis, he said that unit factors, or in other words, alleles, they segregate randomly. They do not mix or contaminate each other. So when sperms and eggs are formed, one of each of these allelic pairs gets randomly distributed to each gamete, and the outcome can be predicted statistically. He gave the example of how when a capital D small d heterozygous plant makes pollen or eggs, each one can randomly receive the allele, the capital D or the small d allele. So this is a debatable, while segregation takes place on its own, how how random it is something that has been questioned in the modern genetics, and that is again an advanced concept that we get to. But just based on what Mendel had said, he said that each of these traits, which gamete will get which trait, that occurs in a completely independent or completely random manner. Not independent. Independent comes in the next law of Mendel, but he said that it occurs absolutely randomly. Then he said in this fourth proposal or fourth concept that was brought forth by him, which is for independent assortment, he said that during this process where a gamete is being formed, the segregating pairs of unit factors, they assort independently. Independent assortment is a concept that has a lot of ifs and buts associated with it. He said that two alleles, when they are getting segregated at one particular locus, for example, like I gave the example of hair color located at P14, he said that when a cell is dividing, and when it is not dividing, when gametes are being formed, during that process, it is a process of division only, it's meiosis which is taking place. So when that segregation of chromosomes is occurring, P14 ka segregation has no effect on the segregation of two alleles at another locus unless linked. This came about much later. So this actually brought forth the concept of linked genes, which showed that in a large number of instances, the segregation of alleles at one locus is indeed impacted by the segregation at another locus because they are linked to each other. They might be located, physically located very far away from each other on the chromosome map, but they can influence each other and they can influence how the daughter generation or how the upcoming generations of progeny would be able to inherit these genes. And therefore, some criteria or some characteristics in a large number of individuals are very linked. Cannot give you any real life examples of it because I can't recall them at the moment. But say, take another example. Say your hair color and somebody's eye color. Say for it never happens. I'm just giving you a random example. But say somebody say on in a particular species, hair color and eye color are linked to each other. If that is the case, you will see that whatever parental combination was there, whatever hair color the mother had and the eye color mother had versus what the father had for each of these traits, you will always see that this pattern is being retained in the progeny. Either it is inherited as a set or there is something else which is occurring in them, which is resulting in a mixed trait. There comes another concept of recombination, again, a very detailed concept. Let's not go into it, but just for now, keep in mind that there are some characteristics or certain traits in individuals which are linked, which is because the inheritance of the genes or the alleles which encode for them, they are linked in nature. That information is good enough. So yeah, these were the ideas that were brought forth by Mendel. Now he said that if we have to correlate what was initially proposed by Mendel and what is known in the modern terminology, then he said that unit factors, they occur in pairs. So equivalent to that are individual genes which are present on homologous chromosomes that occur in pairs. Second was the segregation of unit factors during the process of gamete formation. So on homologous chromosomes, genes, they segregate during the process of meiosis, like I just discussed. Then came the idea of independent assortment, which is for segregating unit factors. That is on independent assortment is seen for non-homologous chromosomes. Say, for example, um, say chromosome 1 and chromosome 15. If they assort independently of each other, then the alleles which are present on chromosome 1 will not influence those on chromosome 15 and vice versa. So in such circumstances, you can see completely independent assortment. And that is where Mendelian genetics comes true, even for eukaryotic organisms and even for certain traits which are more complex and more difficult to study for higher organisms. So you will find instances where each of these hypotheses may or may not be true. Um, then came that the stronger evidence from for the chromosomal theory of heredity, they came from the experiments of Morgans and other fruit flies, which came much later from 1900 onwards. These were way earlier if you talk about the concepts. Gregor Mendel gave his results like we've already discussed just based on what he could see. But certainly experiments over time and detailed experiments over time have made it more possible to understand in much greater depth what is occurring. So you can look at Morgan's experiments for your reference, but obviously it will complicate things for you if you're not familiar with genetics. Um, yeah, because the reason why I'm saying that is because he looked at he looked at different traits like, wing color, wing shape, eye color. There are extensive experiments with Drosophila which have unraveled a lot more about genetics that has obviously helped us, help the scientific community in the long run, but they might be a little too complicated for this course. So you can look it up just for your interest if you want. Um, Now coming to the idea of segregation and independent assortment, like we talked about here, you are seeing two different chromosomes, these homologous pairs of chromosomes, and you can see that different loci within these chromosomes. Sorry. Different loci between of these chromosomes, they encode for different things. Like for example, you can see that the color of the flowers, the white flowers here, like for example, on this chromosome, you can see that it encodes for white flowers, whereas the pair of allele or the allelic pair which is present on the other chromosome encodes for purple flowers, or it might be some other gene as well. Similarly, you can see that how different chromosomes, they have distinct regions, or in terms of genetics, distinct loci which encode for distinct traits. So you have flower color, seed color, flower position, pod color, seed shape, pod shape, plant height, each of these being encoded in different regions within different portions of the chromosomes, or different portions of different chromosomes, let me put it that way. So what you need to understand is that initial experiments of Mendel were all, or initial conclusions that were drawn by Mendel were all based on monohybrid crosses, where you have two different or you have pure breed plants. Like I explained initially, they are plants that have been bred over generations for some particular traits, and therefore they are exactly the same in all traits, except they vary, or they are different or variable with respect to one single trait, and they are crossed with respect to each other. So the first generation, which are being crossed to each other, is known as the parental or P generation. The progeny which are a result of this particular cross is known as the F1 generation, or the first filial generation. And then the subsequent generations are F2, F3, and so on and so forth. So what you see here are, for example, the parental generations. They are pure breed plants which only vary in one particular trait. That is, they either have smooth seeds or they have wrinkled seeds, and these are crossed with each other. And in the F1 generation, we are seeing that the progeny, yeah, in the subsequent generation, or in the F1 generation, we are seeing that the progeny, they have all smooth seeds, from which you understand that among these two traits, smooth, the alleles which code for, or the unit factors which encode for smooth seeds are dominant in nature, which is why you are able to see that in the F1 generation. So these were simple but very remarkable conclusions that could be drawn from monohybrid process. So these are between strains that have variants or that differ in only a single trait. However, another particular concept which is explored is reciprocal crosses, which is where you want to understand if a trait depends upon the sex of an organism. So you will reverse the gender of the plants that are being crossed. You would take a female smooth seed versus a male wrinkled seed, and vice versa, and see whether it is being retained on. The reason why it is important to do that is to understand whether that particular character that you're looking at, if it is encoded on autosomes or if it is encoded on sex chromosomes, because based on that the inheritance would also vary. So there are certain traits which are not just for pea plants, I mean for humans as well, which are encoded on particular chromosomes, and based on that their presence within an individual can vary, and therefore certainly their inheritance can also vary. If I give you one particular example off the top of my head, the Y chromosome, for example, has often been linked to hypertrichosis of pinna, which basically means if you see in a lot of males, especially after a certain age, you will see that their outer ear, the part, the pinna that you talk about, uh, how do I say, the cartilaginous part that you can touch, that has a lot of unnaturally is increased hair growth. Now, while under normal circumstances, if a particular region within the body suddenly shows a lot of hair growth, it is something which is considered to be worrisome. Can often be linked to melanoma as well, not in case of Indians very frequently, because we have darker skin tones, we are more, we're much richer when it comes to the melanin content in our cells. So there's a lot more protection. But in the West, majorly, you can see that the population that has a lot of, which has reduced melanin, which are very sensitive to sun exposure, they often treat this with a lot more seriousness. But compare that to the simple overgrowth of hair, or if you can think about pattern baldness in a lot of men, you often see that fathers, when they are bald in a large number of cases, their children, it's certainly not an easy link to draw, but you can see that there is a large proportion of population where parents, or rather the male parent, when they have a certain pattern baldness or they have a hairline issue, more often than not, their progeny suffers from the same, because certain characteristics, they are being retained on the Y chromosome, as a result of which Y-linked inheritance can be seen in those individuals. So you can see that across generations, it will only be there in males. Or you can see that if it is an X-linked trait, then this you would see that it would only be present in, say, for example, certain diseases like hemophilia. These are X-linked diseases. There are other examples of this also. But if you see that in a particular generation, a father has hemophilia, and in the next generation, you're only able to see it uh, in the girl child, in the female child, that means it's an X-linked trait, because a father will never contribute his X chromosome to his son. He would contribute the Y chromosome, whereas the mother contributes one X chromosome and the father contributes one X chromosome to the girl child, and therefore you see an X-linked inheritance there. So all of this can be studied in a lot of detail, and you can understand how you understand the genetics of certain conditions, certain diseases, certain traits based on just studying the way in which it is being inherited across generations. So certainly you can draw your own conclusions. Mendel did this with just pea plants. So take a moment to appreciate how remarkable his discoveries were. So basically what we see here in this last slide is the way in which the characteristic of tallness is inherited across generations. So you see that in the parental generation, there's one tall, purebred tall plant, which is represented as capital T, capital T, and there's a purebred short plant, which is represented as small t, small t. So the reason why you take purebred plants is because with respect to these characters, you would only expect one type of gametes to be produced from some such plants, right? You would only get a capital T allele, and here you would only get a small t allele, and therefore in the F1 generation, you would certainly expect that there would only be a heterozygous population, because they need two copies of that particular um, trait or that particular locus. Therefore, they would have a combination of each of these. So you would have capital T and small t in that case. Then two such plants, when they are mated with each other and they are mated in a combination where one of them is male, one of them is female, therefore the gametes which would be present would also be the same in each case. So you would have one capital T versus one small t, and similar for one capital T and one small t for sperms. Sorry, you would expect that the progeny which is produced out of them would be genotypically in a 1:2:1 ratio. You can see it in the representation here as well, that the F1 generation would have capital T as 1 to two populations, or the probability of occurrence of capital T small t would be 2 out of 4, whereas only 1 out of 4 population would be small t, which are purebred. However, the phenotypic ratio would be such that it would have it could have 3 tall plants, whereas only 1 out of 4 possibility of having a dwarf or a short plant. So this is basically the idea that you get from monohybrid crosses, that you can understand what are the possible genotypic and phenotypic, sorry, what are the possible genotypic and phenotypic ratios which could be observed, or which would be observed in the subsequent generations. So that is what we look at till here. And in the final lecture, what we basically understand is the link between what Mendel had seen and or what Mendel had hypothesized versus what is the modern idea. So for that, let us draw like a holistic review of what Mendel had proposed. So you can see here that these are some of the common ideas that Mendel had suggested during the 1860s, where he first said that for him, according to Mendelian genetics, one particular allele completely dominates the other. So one is the dominant trait, the other is the masked or the recessive trait. Second, he said that all genes have two allelic forms. So When you talk about the current situation, these are quite different. So we do not necessarily see that there has to be one allele which is dominant over the other. Secondly, we do not necessarily see that all genes only have two allelic forms. There are many genes which have multiple alleles. Example, skin color is often seen to be a result of multiple allelism. Then we have all traits which are monogenic. He said that all traits are monogenic and are affected by only one locus. Again, as you already understand, this in the current scenario is understood that is not the case. You have multiple loci which encode for this. I don't know. Multiple loci which can actually encode for one particular trait. And next he said that all genes are sort independently. Again, not always true. They are true for certain genes, but not always true. You will see that there is some sort of independent assortment. While might be true in a large number of cases, linked genes are a concept that is extensively being explored even today. And lastly, he said that each trait is controlled by a different set of factors. Uh, Mostly it is true, but it is also possible that the same gene can actually influence multiple traits. Again, the concept of degeneracy, like we saw for the genetic code, that comes in here. So it depends. Again, it's very context dependent. We can't really make a generic statement about it right now. So that was the idea behind Mendelian genetics. Now, coming to what Darwin had said, Charles Darwin, he basically brought forth the idea of evolution somewhere around the same time. And while Darwin had like his ideas were a lot more accepted than Mendel's were at that point of time, it still took decades to finally, bridge the gap between the two and come up with a hypothesis about what is relevant regarding genetics in the current scenario and how it can be verified or validated using the current technology and experiments that we have today. So, according to Darwin, he said that phenotypic variation it exists between individuals. It is why you and I and everybody around us we are phenotypically different from each other. However, it might be looks, height, skin color, hair color, eyes. Just overall, we look different, but we have some similarities within the same family, which is why he gave the idea of heritability that whatever characters we have, some amount of it is being retained across generations, which is why some phenotypes are similar, but not all. Then he said that those individuals that have heritable traits that are better suited to the environment would survive, which as it's very commonly and a little loosely used in the current scenario that Darwin talked about survival of the fittest. He did bring about that concept, but it's not as generic as it is made to sound. um, the basic idea was that if you have the kind of characters that allow you to adapt to the environment that you are in. So, for example, there is a concept, if you ever get time to read about it, read up on founder effect and founder population. It's very interesting. Say, for example, you think of a family think of a student, a PhD student who has moved abroad, who is the first in his entire line of families to move outside India and to settle, say, somewhere in New Zealand. And they have completely different environment. They completely different cycle of seasons that occurs in the entire year. And every living condition that you can think of possibly is different there. So if the first person who goes there, if they are able to adapt, and if they live there in that condition, and they are, they marry somebody who is from there, and they are producing children who are growing up in that very environment, what you expect is that the children should inherit whatever characteristics their parent has adapted over time to survive better in those conditions. If they have, say, for example, four children, of which two are better suited, or two of them have adapted the characters that allow them to survive better in New Zealand, versus two which have not done so, obviously the first two would outlive the other two, because you would expect the other two to be more susceptible to whatever environmental changes are occurring, whatever diseases are more prone there, or they might suffer from hypothermia, they might not be able to survive well in that environment. They might fall sick easily. So a number of things can happen. That is basically the idea that Darwin had tried to convey, that the better you adapt, the better you inherit whatever characters your parent had to adapt to the environment that you are now being subjected to, the better you have the chances of surviving. That was the idea. It was with the perspective of evolution. Versus what Darwin had explored were different. They were with respect to just genetics. So bridging these two were certainly a difficult task. So let us come here. Somewhat these concepts, they were bridged by these group of scientists that you see here. There is Ernst Mayr, who had a role to play there. Then there is Stebbins, Simpsons, and Dobzhansky, who had kind of tried to somewhat understand how population genetics works, and they had tried to bridge these concepts. And what effectively they did, while it is not the full and final version, and as you can see, this was also a really, really long time ago. This was somewhere in the 1940s, and we are almost 80, 85 years past it. So currently the scenario is way more evolved, especially considering that we have so much sequencing data that is available. We have the human genome sequenced. Till now, if you are familiar with it, around I think 2 weeks ago, even the X chromosome got sequenced for the first time, which was a huge mystery to mankind for a really long time now, because the Human Genome Project was started in the early 2000s. And while a lot of information was available, a lot still was unrevealed. So basically, what I'm trying to say is that with evolution, with time, when I say evolution, I mean with technological evolution over time, more and more details would be understood. But back then, what they tried to do was that they sort of combined the idea of Darwinian selection, which is, if we put it shortly in, just briefly as one concept, it is survival of the fittest. They tried to combine that with Mendelian inheritance, that this is how inheritance is carried out, and they tried to propose a sort of amalgamation of the two. And they sort of explained what is population genetics. They studied the genetic variation within a population, and they emphasized on characters. And how I talked about certain characters which are just influenced by genetics, some are influenced by environment, etc. So how those could be understood is because some characters are quantitative. Like for example, height, you can measure a population of, say, a class of 50 students. All of their heights can be measured. That is a quantitative trait. If you try to measure somebody's skin color, you can't measure that. You will get a gradient or a range. Um, and that is because there is not one gene, or there is not even a set of loci which can be associated. There is a large set of loci, and there's a lot more, like I said, environmental characters. Say somebody is more exposed to the sun, they would be more tan, and you wouldn't actually know what is their skin color. Somebody might suffer from a disease, which is why their skin color is affected. Somebody might have vitiligo, which is a disease. It can be a number of other conditions. So some traits are easier to study, some are not. But yeah, you can classify them as quantitative and qualitative traits, and you can get some sort of idea as to how they might be inherited across generations. So this is what the concept of modern synthesis, taken up by these eminent scientists, kind of explored. Then came the idea of non-Mendelian inheritance. So basically what we are trying to understand is that Mendelian genetics, like I said, proposed a certain concept. Non-Mendelian genetics deviated from the same. So Mendel had made his conclusions based on just the pea plant that he studied. And later, like I said, there was a lot of deviation from what he had suggested. And therefore, his ideas had to be rephrased or modified to understand in much more depth. So from there came the diverged concepts of incomplete dominance, codominance, penetrance, sex-linked inheritance, dosage compensation, sex determination, epigenetics, multiple allelism, lethal alleles, epistasis, pleiotropy, expressivity, and genomic imprinting. Now, these terminologies can be a little bit scary, but these are very, very deep, like these are deep topics. If you want details, I can give you some idea about it, but I would suggest you don't go into it unless explored in much detail. Mainly what you can look at are incomplete dominance, codominance, and multiple alleles. And these I think you'll find these even in senior secondary books. Sex-linked inheritance I have introduced to some extent. Dosage compensation, again, a little more complicated. Um, and is kind of related to what epigenetics also is. Lethal alleles, epistasis, pleiotropy, these are much more advanced genetics. So we'll steer off of that for now. Then came the idea of horizontal versus vertical genetics. What you see here is an idea of vertical genetics, which is from parental generation to the next generation, like the example that was given in the lectures as well of Amitabh Bachchan, Jai Bachchan, and their son Abhishek Bachchan, and how you can see that the genes which were present in the parents, what part of that is getting carried to the next generation. Versus if we talk about horizontal gene transfer or horizontal genetics, it is, if I put it very simply, it is not familial or it is not within a family. There is some external factor which is influencing the genetics of a particular individual. Say, for example, say if I get, somehow I get infected by a virus or okay, let's take the example of a virus only. A sort of viral pathogen which comes and integrates its genome into my cells, some of my cells. What will happen is that my parents did not get infected by this particular virus, neither did their grandparents, so that is not a familial trait. So normally I would not be expected to transfer that to my future generations as well. But if under certain circumstances, for whatever reasons, this particular viral genome, it gets integrated somehow into my germ cells, which are the gametes, which will get transferred to the next generation, then this would become a trait that gets transferred over and over and over generations. It should not happen because there is a lot that your body does to make sure that even if you get mutations, you do not get them in your germ cells. But there are exceptions to this, so you never know. But yes, the order in which this transmission is occurring, if it is directly from parent to child directly, then it is vertical gene transfer, versus if it is because of some external influence which is occurring suddenly in a particular generation, then that is horizontal gene transfer. So that is what we see here. And here you see post modern synthesis, the concept that was brought forth. These were all the different topics that were brought about. I'm not getting into details of this. And here you can see how evolutionary how evolutionary biology has taken all of these different concepts into consideration: Mendelian genetics, population genetics, evolution ecology, along with other topics as well. So these are just for your information, but you don't need to get into details about this. Segregation and independent assortment we have looked at. We understood the dominance with respect to tall traits. Then came the idea of Muller's morphs. So morphs basically mean phenotype. You can think of it as equivalent to a phenotype, what you can see basically. So what he tried to explain was why are some mutations dominant, why are others recessive. So if you look here, this is a wild type allele. This is a representation of a wild type allele. What is it producing? It is producing a functional protein, which is giving you the wild type phenotype. When there is a loss of function mutation, a loss of function mutation basically means that that region is lost. As a result, the allele which was present is producing a sort of a variant or a version of that original protein which is no longer functional. So what that would lead to is that it is a, so this variant can also vary, right? It sounds confusing, but basically what I'm trying to say is the type of mutation or the type of change that has occurred, that is not very absolute. Either it can be that, say, a particular region of the genome which was encoding for one particular protein, it's deleted and it's completely lost. If that happens, what I will see is a very severely mutated phenotype, and that condition is called as an amorphic loss of function. That because this gene is no longer present, it has been completely removed. What I see is an amorphic condition. Versus if I see that there is some sort of a mutation which is changing or altering that gene in such a way, which you can see here as well, that here you get some parts of it which are similar to its original content or original shape or function, then it is somewhat able to perform its function, but not to the best of its abilities. So what you see under certain condition is that the mutant condition is there, but it is quite mild. So it is a hypomorphic loss of function. So the like it was explained in the lecture as well, if you think of it as a scale, right? If the normal function or the normal morphism is a wild type morphism is 100, then with respect to that, a loss of function condition is it's completely gone. There is no, you can't really compare it. Versus if it is say 70, 60, 50, which means it is less, but it is still there, then you call it to be a hypomorphic function. Versus if there is a certain condition where there is a different allele, a dominant negative allele, which you see here represented as AB, which produces a different polypeptide that can interfere with the wild type polypeptide, which means that you have the normal protein in that individual, but you also have some sort of a controlling protein, which can somehow interrupt or interfere with the function of the normal protein, then that also leads to a very severely mutant phenotype, and that condition would be known as an antimorph. So it goes against normal function. So despite the fact that there is one particular allele or one particular unit factor which is producing the normal protein, there is another allele which is producing something which is stopping the first one and therefore is inhibiting its function. So that is what we see are different types of morphs that were suggested by Muller. So yeah, so here to see another representation of how dominance might work. So you have the genotype where it can either be homozygous or heterozygous. This is homozygous dominant, capital S for smooth seeds, which we talked about previously. This is smallest represented by purple for homozygous recessive, which leads to wrinkled seeds, versus you have heterozygous condition, which also leads to smooth seeds because the capital S allele, it dominates over the small s allele and masks its function, producing smooth seeds. This explains the concept of dominance. Then we talked about the monohybrid cross before, and these are the ideas of purebred lines and how they are generated. So you have the parental generation, which has smooth seeds, and cross them with plants having wrinkled seeds. You get F1 generation, which has all smooth seeds. You self-cross them, that is two smooth seeded plants are crossed with each other. In the F2, you will get a much higher population of smooth seeds, but you still see that you would get some plants which would have recessive genes. So there is a mystery of this. We are not getting into details of this, but basically the way in which genetics are studied are, or commonly studied, are with respect to a Punnett square, where you place the female gametes along one side and the male gametes along the other, and you get the combination of these zygotes within the squares. So I think these were not there in the lecture, but yeah, I think we can skip these because these were not part of the lecture. But you can get into it as well because we have some time. Okay, so monohybrid crosses here, not much has changed. Basically, the representation is being done with respect to a gene. So again, the smooth seeds are being represented, as you can see, with S. So this S is here. It is representing the enzyme SEBI, which is a starch, SEB1, which is starch branching enzyme 1. What it does is it synthesizes amylopectin, which is a sort of a compound that is responsible for the structure of the outer coat of the seed. So round or smooth seeds, they contain amylopectin, and they shrink regularly when they lose water, while the wrinkled ones, they lose water quite irregularly. So the expression of amylopectin determines how the seed would look like on its exterior. So that is what we see here. That with with respect to the gene that you look at, you can understand how exactly it is getting influenced. And the same thing has been explored across two generations using the Punnett square as well. So you're crossing smooth seeded plants with with wrinkled seeded plants. And in the F1 generation, you are getting the phenotype that all of them are smooth because it is capital S, small s. And you are able to see that irrespective of the genotype, the phenotype is the same. So all phenotypes are smooth and all genotypes are capital S, small s. Versus when you cross these plants with respect to each other, which are capital S, small s, you see that their gametes are different. You get a mixture of gametes, as a result of which you will get a phenotype, but 3 out of 4 of these plants are smooth, whereas 1 out of 4 of them are wrinkled, and 1 out of 4 of them only has the homozygous dominant phenotype, which is capital S, small s. So basically, the Punnett square is used to understand that. And also, he showed that Mendel had showed that all of the different traits that he had studied, they showed similar behavior. So he differed one of the 7 characters in each case. Like you see the you see the process for the smooth seed coat, the color, the flowers, the pods, the stems, etc. So he looked at all of them, and this is with respect to humans, how earlobe, whether they are attached to the person's face or unattached to it, that inheritance can also be studied. Then here you see an example of a dihybrid cross. I think I'll not get into it today, but if you have doubts, you can mention it. We'll get into it in the next class. But yeah, it's slightly complicated, but you can go into it because it was not there in the lectures. I'm not delving into it. But if you have doubt, please bring it up in the next class. Why people are joining now? But basically, what I will switch to now are some sample questions for the next week's assignment before I end the class for today. So, joining very late. Okay. So we were looking at sample questions. Which of the following is not explained by Mendel's laws of genetics? So if you think about the different laws that he had proposed, there was the law of dominance, the law of segregation, and the law of independent assortment. He does not delve into the concept of linkage. While it was mentioned that Mendel talks about the possibility of independent assortment only if the genes are not linked to each other, he does not really go into that concept or does not provide an explanation for it. So that is what we see there. Then in question 2, a mutant that produces more proteins or more active proteins than the wild type would be called if you've listened to the lectures, this is a direct question from there. That is a hypermorph. So an amorph is something that has a complete loss of function. A hypomorph has less than normal function. Hypermorph has much higher function than wild type, and antimorph has something which is impeding the normal function. Then in Mendelian genetics, if two heterozygous parents are crossed with each other, the phenotypic ratio of the offsprings is. So make sure you read the question carefully. You are talking about heterozygous parents. So that would be, if you take the example of tallness, then it is capital T crossed with capital D. The gametes would either be capital T or, capital D small t with capital D small t. So the gametes in that case would either be capital D or would either be small t. So if you make a Punnett square out of those, you will get a genotypic ratio of 1:2:1, but a phenotypic ratio of 3:1, which means that 3 out of those 4 plants would be expected to be tall. So that is the answer to this question. Next, the idea that different pairs of alleles are passed to offsprings independently is Mendel's principle of independent assortment, as you can directly see here. Do not get confused between segregation and independent assortment. This particular concept where he said that to the offspring they get passed independently, that he talked about independent assortment. Whereas the segregation, he said that from the parent to each of their gametes, the segregation is completely random. So there is a fine difference between the two, but you can look into those as well. Then another question is talk about how a plant with heterozygous alleles has blue flowers and one of the alleles codes for a white flower color. Which of the following statement is true? You have a plant, it is heterozygous in nature, and it has blue flowers, which means that the trait that you are able to see in the phenotype is the blue color, versus you are also understanding that it has an allele that, because it is heterozygous in nature, it has one of the alleles that it has one of the alleles coding for a white flower color as well. So it has both, but blue is the one which you are able to see. So which of the following conclusions can you draw from it? The first is that blue is a dominant trait, white is a recessive trait, blue is recessive, or both A and B. The correct option is D, which means that yes, blue is a dominant trait, but also white is a recessive trait. So make sure you read all your options, because there's a high chance you would just look at the correct option for option one and leave the question as it is, but that would not be true. White, it's also, if you are considering one trait to be dominant, you would expect here, using Mendelian genetics, that the other trait would be recessive to the dominant trait. So option D would be correct in this scenario. Then in question 6, we are being asked which of the following statements about enzymes is not true. Um, all enzymes are proteins. Enzymes reduce the activation energy of a reaction. They participate in cellular metabolic processes and they speed up a reaction. So obviously option 4 is correct. They do speed up a reaction. That is literally their job. They do participate in cellular metabolic processes because it is necessary for those processes to be done in real time as they are. Otherwise, it would take hundreds and thousands of years for one particular process to be executed. Third is that enzymes reduce the activation energy of a reaction. They reduce the energy that is required to reach that intermediate state. So that is their job. The first option that you see here, which is that all enzymes are proteins, while it might look logical, it's not always the case. Why are we saying so? Because in a large number of cases, and one which you are already familiar with, RNA can also act as enzyme. So we talked about, like during translation, we talked about how the ribosome is a ribozyme, right? So you don't have, like, while the ribosome is an assembly of both RNA and proteins, it has been studied and studied extensively that the catalytic part of a ribosome, weirdly enough, is the RNA and not the protein part of it. So RNA is capable of functioning as an enzyme. And that is one of the reasons why the concept, another thing for you to look forward to if you're interested, that is one of the reasons why the concept of RNA world, or the RNA world hypothesis, was brought about. That from the from ancient times, what existed was RNA, and from there we have evolved to DNA being the master molecule. So another concept you can look at. Um, in 7, we are being asked which of the following is the most abundant biomolecule found on Earth. So while you would expect it to be proteins, they are carbohydrates. Carbohydrates are the maximum, they form the maximum proportion of biomolecules on Earth. And one way you can think about it is, if you think of which living beings are the most predominant, they are plants, and plants are extremely rich in carbohydrates, because if you think about it, what is their main function? What is the main function that you study with respect to plants? So carbohydrates form the majority of car, not carbohydrates. Plants, they form the majority of the contributors towards carbohydrates in the living world, or on Earth in general, and that kind of justifies the answer to this question. Um, then comes question 8, where a test cross is performed to determine the genotype of a tall plant. The cross results in an equal number of short and tall plants. So the genotype of a parental plant is, so this you can, if you just go through the lectures, you'll be able to see it. You'll see that a 1:1 ratio of tallest to short, it comes when the plant population is heterozygous in nature. You can just look up any test cross for your understanding. So the objective of a test cross is exactly to do that, to understand whether the population that you have gotten, say, for example, if you have a tall plant, whether that tall plant is capital T or capital T small t, that you understand by crossing it to the homozygous recessive parent. That is the test cross, because you're testing to see what is the genotype of the plant that I am interested in, or of the individual that I am interested in. So that is how test cross is basically performed. It is quite simple to do. You will see that you'll get a 1:1 ratio. If you face trouble, please let me know and we'll take it up in the next class. Um, in question 9, we are being given an example of a dihybrid cross. I did not get into it today, so I don't think I'll expand much on it. Again, go through it. I'll read out the question anyway. In pea plants, seed shape can be round or wrinkled, while the seed color can be yellow or green. Round is dominated over wrinkle, or yellow is dominant over green. So in a cross between capital R, small y, and capital R, small y parents, how many offsprings will have round and yellow seeds? Okay, so I think we can go into this without even getting into much detail. Basically, you just have to look into the fact that this statement is very, very important. We're being told that we are being told that round dominates over wrinkle and yellow dominates over green, right? So capital R dominates over W, and I think it should be small r, but yeah. I think the representation here is not correct. Whereas yellow, it dominates over green. So small y dominates over small y. So here you are taking two parents who are of which one has round seeds, the other phenotypically has round seeds but is heterozygous for the same. And you are taking both parents which are both green in color and they are not yellow, right? So the question that has been asked is that how many offsprings will have round and yellow seeds? So obviously, if you just look at just the color of the seed, you will see that none of the parents have the capital Y gene, right? Or capital Y allele, right? So neither of them can have yellow seeds. So this is a direct question. You don't need to go into Punnett square, etc. They do not, neither of the parents will give the gamete via which you can get a plant with a yellow seed. So the answer directly becomes 0. That's why I went into this question. But if you want dihybrid cross to be explained in more detail, please bring it up. We'll take it again in the next class. And the last question is that a mutant allele where the protein product binds to the wild type protein and inactivates it is a amorph, hypomorph, hypermorph, or an antimorph. So we have discussed this as part of Muller's morph as well. So an antimorph is the condition where you have a mutant allele which encodes for the kind of protein which goes ahead and inactivates the normal protein and interferes or interrupts its function, interferes with the function of the normal protein. So that is the concept of an antimorph. So I think that kind of covers everything that we had to take up for today. I did rush through a couple of topics, but in case more detail is required regarding the same, you can let me know. And apart from that, if anybody has any doubts, please go ahead. Okay, if not, then thank you for joining. You can submit your questions if you have any in the discussion forum as well. They are addressed by a different official TA for the course, so maybe they'll get into it. Or you can ask in the next session or any of the other discussion sessions as well. So all the best for your assignment for this week, and we will meet again on next Sunday. So thank you so much, and good evening and bye.